Transcription is the process where RNA is produced by the reading of DNA. RNA is used to produce proteins within cells. In this video, I represented the transcription process by baking a cake and using icing on it. Transcription begins at the promoter region where RNA polymerase binds to the DNA. RNA polymerase binds to the DNA at the -35 region as a hollow enzyme, including the sigma factor, which later dissociates after about 9 to 12 nucleotides have been read. When the sigma factor dissociates, the core enzyme is left. The -10 box and +1 regions are also very significant in the transcription process. Both the -35 and -10 regions are known as consensus sequences. The +1 region is where elongation begins and base pairs are added. Once the sigma factor dissociates, elongation begins. In this example, transcription is occurring towards the right. The core enzyme binds ribonucleoside triphosphates by pairing the bases with the template strand of DNA. The nucleotides are added at the 3' end with phosphodiester bonds to the last nucleotide. The corresponding bases will be added, like in the double-strand model of DNA. However, the bases of RNA include guanine, cytosine, uracil, and adenine. Thymine in DNA is replaced with uracil and RNA, making uracil and adenine a pair. The template strand of DNA is what RNA polymerase reads to add new nucleotides. The other strand is known as the coding strand and will be identical to the new RNA strand, besides where thymine is replaced with uracil. When the transcription process is coming to an end for a specific RNA sequence, termination must occur. One way that termination can occur is with the involvement of the protein Rho. Rho will bind to the RNA, later reach the RNA polymerase, and cause the DNA and RNA to unwind. The other way that termination can occur is by the formation of an RNA hairpin loop. The hairpin loop is the binding of a region of the RNA strand with itself. The adenine and uracil bonds are relatively weak and cause the DNA and RNA to dissociate. If there was an insertion of 5 base pairs between the -35 region and the -10 box of the gene listed in the prompt, the organism would have major repercussions due to production of phenylalanine. The promoter region, which is where the base pairs are inserted, determine how often the gene is transcribed. With the insertion, this would be inaccurate and could potentially cause an increase or decrease in the amount of phenylalanine. Based on how important this amino acid is for the organism would determine the effects that the insertion would have. However, it would likely greatly influence the organism due to an altered production of the amino acid. All of the information used to create this video was found in the Module 4 lecture slides for this class. All of the figures were created by myself.